Okay, hi everyone. If any of you don't know me, I'm Ben, and I'm going to be talking today about mechanical signaling in the meniscus, or mechanotransduction, which is the translation of mechanical signals into biological signals in the meniscus. And for the past year, I've been working on developing some tools and methods to study this. I'll present some results from that and talk about some of our planned experiments to study mechanotransduction in the meniscus and why we think this might give us some clues as to how we can improve outcomes for people who have had meniscus injuries. So first of all, what is the meniscus? Or menisci plural are C-shaped discs of fibrocartilage. They sit on the tibial plateau and form kind of a socket for the femoral condyles of the knee. And along with cartilage, they function as kind of shock absorbers of the knee, and they're also very important in stabilizing the knee joint. As I said, they form sort of a socket for the condyles, and along with the ACL, they're kind of the primary structure that really stabilizes the knee joint. So here's a top-down view showing the medial and lateral meniscus, and I've drawn kind of a line separating the outer and inner zones because they are somewhat different. Most obviously, the outer third is vascular. The inner two-thirds approximately is completely avascular. There's also some differences in the composition between the inner and outer zones. The outer zone is primarily type 1 collagen arranged in longitudinal fibers, where the inner zone is a little more like cartilage, and it's primarily type 2 collagen and has pretty high proteoglycan content. And this allows it to kind of have that shock absorber function because the proteoglycans are negatively charged, creates an osmotic gradient that holds fluid in the tissue, and then when it's compressed, the fluid is forced out and it's drawn back in by the negatively charged proteoglycans. So the meniscus is primarily composed of extracellular matrix, but it is not acellular. There are cells present, and we refer to them as meniscal fibrochondrocytes because they have some of the properties of a fibroblast and some of the properties of a chondrocyte. And beside the endothelial cells in the outer zone, obviously, it's really the only type of cell that's found in the meniscus, although there is some phenotypic differences between the outer and inner zone cells. The inner zone are a little more chondrocyte-like, where the outer zone is a little more fibroblast-like. But these phenotypes are kind of poorly defined, and you'll see that kind of come up later in the talk, that it would be really useful if we had some more meniscus-specific markers, as well as a better idea of exactly how different the inner and the outer zone cells are, and what that really means. So meniscus injury is really very common. One study, which they calculated kind of an injury rate based on emergency room visits in a certain city, and then based on the population of that city, they were able to calculate sort of a population risk. And they measured a rate of 0.7 per 1,000 people per year, and I would say that the actual number is almost certainly higher because not everybody who has meniscus injury is going to be seen in the emergency room. And I just like to kind of compare this to ACL injuries because people are kind of aware of ACL injuries because of high-profile athletes who often have these injuries. And so just for comparison, they measured a rate of 0.3 per 1,000 people per year for ACL injuries. So meniscus injury is at least twice as common. And actually, meniscus injury often occurs at the same time as ACL injury, and several studies have found that when this is the case, the degree of damage to the meniscus is often the most important prognostic factor. So, as I mentioned, the importance of the meniscus in stabilizing the joint along with the ACL is increasingly being appreciated. So the symptoms of a meniscus injury would be pain, swelling, and stiffness. But what really causes people to seek treatment is that the knee can catch or lock up. And what happens, you can see there's sort of a flap or bucket handle type tear It's really usually pretty much asymptomatic if the damaged part is like in place, but if the flap becomes dislodged or moves out of place, that's what causes the knee to lock up, and it's very painful. So I'd also like to point out, just again comparing to ACL injuries, which are almost entirely sports related, that meniscus injuries, while they are commonly caused by sports activities, they can also be attributed to a variety of different reasons. So they're really common among the general population, not only athletes. And I don't have the data here, but they're also much more common throughout life, where ACL injuries are pretty much only seen in young people. Meniscus injuries probably peak more in middle age, where you have both maybe some age-related degenerative changes, as well as maybe still active enough to have acute type injuries. So treatment of meniscus injury in cases that are symptomatic, the treatment pretty much is either a meniscectomy, so remove the damaged tissue, or in some cases it can be repaired. And partial meniscectomy is probably actually the most common or commonly performed orthopedic surgery. And what's done is they just remove the tissue, the damaged tissue, and trim it back to a stable rim. Some meniscus injuries can be repaired by suture, but this is pretty much only possible if it's what's called a red-red or red-white tear, meaning that it occurs either in the vascular zone or is partially in the vascularized outer region. So you might ask, does the meniscus really have the capacity to heal? And I would say yes, but not very well. Maybe 60 years ago, the standard of care for symptomatic meniscus injury was actually total meniscectomy, because it was thought to be kind of unimportant. But in maybe the 80s and 90s, the long-term follow-up of these studies of these procedures found that pretty much universally people who had a complete meniscectomy would develop osteoarthritis within 10 or 15 years following the surgery. So since this became, since that link was kind of really firmly established, there's been a push to repair or maintain as much of meniscus tissue as possible. So surgical repair is now recommended for most red-red and red-white tears, and even some are now recommending that radial tears, which are kind of often thought of as not repairable. This is now recommended that repair should be attempted, and they consider a success rate of about 80% to be acceptable, because the alternative is the loss of meniscus function, and this is heavily predisposed to osteoarthritis. This paper found that there is a trend toward more repairs. It doubled over the rate of repair doubled over the course of their study. However, that was still only 6% overall of the surgical cases that they looked at were for repair. So most of the time, meniscectomy is still the preferred treatment, unfortunately. White tears are considered not candidates for repair. Animal models have shown that white tears can heal when they create a vascular access channel, essentially, to the vascular zone, or even just by placing a fibrin clot in the defect is enough. But this approach has not been found to be successful in humans. But I just want to make the point that my takeaway is that the meniscus is capable of healing, but it often does not.
So for the white tears, if they put in some kind of scaffold, will the meniscus material or tissue grow over that scaffold as a repair mechanism?
Yeah, a lot of groups are working on this, including our lab. Has some scaffold materials that we study. So it's an active area of investigation, but there's nothing currently in use that's been very effective. So our lab has focused on the study of mechanotransduction because we think it might give us some clues as to how we could improve outcomes and improve maybe that the success rate for meniscus repair, and maybe make it possible to repair white tears. tears. And the reason that we think this is kind of summed up in this slide. So from kind of a macro scale, we know the knee joint is definitely a use it or lose it organ essentially. And normal levels of physiologic use, such as you might have moderate exercise and walking, have been shown to preserve the joint. The extracellular matrix components generally hold up, are more maintained better in individuals who are active. At a tissue level, it's known that dynamic loading or compression increases anabolic or genes and decreases the expression of catabolic markers. And in this field, people often think of the tissue homeostasis. Most people kind of boil it down to a balance between these anabolic or like the production of extracellular matrix components, collagen and proteoglycans, versus catabolic processes, essentially the production of matrix metalloproteinases that might break down the tissue. So compression is shown to favor anabolism and decrease catabolism in this tissue. And then what I think is maybe one of the most interesting things that we know about mechanotransduction in meniscus and cartilage is that it actually seems to offset some of the negative effects of inflammation. So IL-1 and TNF alpha are kind of some of the most commonly identified cytokines that are elevated in inflammatory conditions in the knee. And studies of loading at the cellular and tissue level have shown that loading actually offsets some of the negative effects of these inflammatory cytokines, which I think is really interesting. So one example of that is this paper, which was published from our lab several years ago, in which, so this is an explant injury model in which we take a biopsy punch of meniscus tissue and then create a smaller punch through the middle, so this is like a full thickness defect. If you put this back, if we put the core back in the ring and place it in culture, the defect will actually repair over time in culture. As you can see here, when we add IL-1 to this system, there's increased MMP, matrix metalloproteinase activity, decrease in proteoglycan content, and it causes a decrease in the shear strength of the repair of the defect as measured by a push-out test. And loading partially or completely offsets this effect.
Question. Do you think that it could have anything to do with increasing perfusion of stuff into the, like, you know, moving things around so that you don't have concentration of whatever it is that's triggering the production of inflammation?
I mean, yeah, that definitely could be the case. I would say that, at least in terms of the normal physiology of the knee, loading causes the tissue to flow in and out, and for these avascular regions especially, that means that all of the nutrient and gas exchange is occurring through synovial fluid. So that could be a factor.
So maybe without strain you have inflammation because you have stasis.
Yeah. It's an interesting thought. I'm not really sure how you would separate out those effects. It might be kind of difficult. So that leads me to the aims of my current and future research, which is to identify some of the transcription programs that are activated in response to mechanical loading. So we plan to perform RNA-seq on loaded and unloaded meniscus tissue to identify genes and pathways that are regulated by mechanotransduction pathways. And then we're also going to do this experiment in the presence or absence of IL-1 to see what sort of IL-1 induced transcripts maybe are modified by loading. So once we have those results, we'll identify some potential genes of interest and do some more confirmatory and eventually some mechanistic studies.
So then what cells make IL-1 in this tissue?
That's a good question. I think that the source is probably from the synovium. I'm not sure if the native meniscus cells might produce a low level of IL-1, but it's probably coming from the synovial fluid, I think is the main. So maybe synovial macrophages or inflammatory cells in the joint
But it's
capsule,
not
but not
recruited.
directly in the.
But not recruited cells.
They don't penetrate into the meniscus. Immune cells have not been known to actually migrate into the tissue. So whatever the source is, I think it's within the joint capsule, but not in the meniscus itself. Okay, so the way that we are planning to load these tissues, we use this custom loading device. So we can take biopsy punches and we try to get them separate, kind of separate out roughly the inner versus the outer zone so we can study them separately. So we can take a biopsy punch and place it in a culture dish and use our loading device, which goes in the incubator. And essentially it's a stage with these pistons. We drop the piston down until it's in contact with the sample, and then the stage moves up and down at a predefined strain rate. And we usually use about 10% strain because we think that's in the range of what the tissue would experience from walking, maybe at a frequency of once a second, 4 hours a day for 3 days in this experiment. And again, we think this kind of approximates the sort of strain that the tissue would experience in regular activity.
My question was 4 hours continuous or 4 hours total throughout the day?
We do 4 hours continuous for these experiments. It would be interesting maybe to see if there's a difference if it's loaded in increments. I'm not sure. Have we ever done anything like that?
Because presumably this loading, you're mimicking, steps, right?
Mhm.
Weight on, weight off, weight on, weight off.
Yeah.
And most people don't do 4 hours a day in a row.
Yeah.
They would do a little bit and then stop, a little bit and stop, and then have a long period of time at night.
Mhm. Yeah, it's true. I think it might be interesting to try multiple shorter bouts of loading and see if there's a different effect. Currently, the way this is set up, I have to manually start and stop every time, so it'd be nice if, with programming, we could really do some sort of a simulated activity throughout the day, but currently it's I go in, start it, stop it. So this part of the experiment actually has been done, and I have samples frozen, but the next challenge is extracting RNA from meniscus tissue, which is what we would call technically challenging, or I would call extremely frustrating, because it's a very tough tissue. It's mostly extracellular matrix. It's a pretty low cellularity, and that means low RNA content. And for this particular experiment, these are pretty small pieces of tissue. It's a 4 millimeter biopsy punch, about 2 millimeters thick. So the method we've been using is to actually hold the sample in forceps and grind it with a high-speed rotary tool, and we try to keep it under liquid nitrogen the whole time to preserve the RNA. And it is not a very fun procedure. I don't think this picture really captures the frustration of what doing this is really like. Like I said here, the one good thing is that it really gives you that kind of mad scientist look and feel when you're grinding a piece of tissue under liquid nitrogen. But the cons are pretty much everything else. It's tedious, difficult, and some samples cooperate, others slip out of the forceps and go flying and get lost, or just won't grind well. So I worked my way through a dozen samples or so with some help from other lab members and sent them off to the sequencing facility, and they ran their QC procedure. And on the left is an example of what this would be high quality RNA. You see the very distinct 18S and 28S peaks. And these are the results we got back. So a couple of the samples met their QC requirements. A few of the samples were kind of marginal, and a few of them failed completely. So I'm currently working on trying to optimize this protocol a little bit, and now we're using a freezer mill, which essentially does the same thing. It pulverizes the tissue while keeping it submerged under liquid nitrogen. And this is automated, so hopefully it will be a little faster and more consistent results. And I just got back the first couple of samples using that method. I just got the QC results back today, and they look pretty good, so I'm looking forward to scaling this up, and we'll hopefully have some interesting results shortly.
Are those 8 separate menisci or 8 separate samples from the same?
8 separate samples. When you set this experiment up, we used 4 menisci and
Inner and outer.
yeah, and we took 4 inner and 4 outer punches from each. So, so this is kind of just a diagram of the whole experiment. And as I said, this part is done. We've taken our inner and outer zone punches. We have those that were subject to compression or control. And yeah.
So the differentiation between inner and outer is just the vascularity. So
Yeah.
one would have blood, the cells from the blood. Is that?
Yeah, there might be a small amount of, there would be some endothelial and blood cells present, I guess. It's something I've kind of wondered about, and I don't really, haven't ever tried to quantify just what percentage are really meniscus cells and what might be blood derived. It's definitely a factor that.
Okay. But that's potentially possible. You would have some endothelial cells contributing to this signal.
Yeah, there would be some amount of endothelial cells there, I suppose, yeah.
So how can you control for that?
You know, I guess I haven't really thought about it very much. I kind of assume that it's not a huge that the primary cell population is the meniscal fibrochondrocytes, but it would be interesting maybe if there's some way we could measure the amount of endothelial versus meniscus cells. If we had markers to do that, that could be interesting to find out if it's really a significant population. So these are from pigs, actually. We get pig joints from a slaughterhouse, and that's what we use for most of our experiments. We occasionally get human tissue from knee replacement subjects, but obviously if they're having a knee replacement, it's not healthy, normal tissue, so it limits what we can do with those samples.
You don't need to look for markers in an RNA-seq will tell you what kind of cells you have.
Yeah, that is something that hopefully the RNA-seq will maybe help answer some of these questions. Yeah. Okay, so a second aim would be to kind of look upstream of these processes and figure out what are the key, really mechanotransduction pathways that are taking this mechanical signal, converting it into some sort of biological signal, and then what we're measuring with RNA-seq would be some sort of transcription program downstream of that. And we'd like to figure out what some of the key players in that process are. So the study of mechanotransduction generally is kind of a new field, and especially in the meniscus, I know of really essentially 2 papers that specifically study mechanotransduction in the meniscus. So we know that the meniscus cells are responding to mechanical load somehow. We really don't know much, if anything, about the mechanisms involved. So this is one of those 2 papers, and they show here that calcium signaling seems to be an important part of this. So here this is measuring intracellular calcium using a fluorescent dye, and the arrow is when they apply some mechanical stimulation, and you see a spike in intracellular calcium. And they also linked this to proteoglycan production. So when they have when the cells are stimulated mechanically, they produce more proteoglycans, and when they use kind of a broad calcium channel blocker, this effect disappears. This effect is also seen in chondrocytes, where it's been studied a bit more. And in chondrocytes, TERP-V4 has been identified as a key mediator of this calcium, mechanically induced calcium signaling. And TERP-V4 is an inducible calcium channel that's known to be mechanically activated in a variety of tissues. So this would allow extracellular calcium to rush into the cell, which gives the kind of calcium spikes that we saw in the previous slide. And in chondrocytes, this is using chondrocytes embedded in agarose and loaded. They show that loading increases proteoglycan and collagen production, and they use GSK205, which is a TERP-V4 inhibitor. This effect disappears. So we believe that TERP-V4 may play a similar role in the meniscus. And I'm planning to test this by knocking down TERP-V4 in meniscus cells and looking to see what effect that has on calcium signaling in meniscus cells, and then to do some similar experiments to what I just showed, where we can embed the cells in a hydrogel and load them with our loading device, and see what effect this has on production of extracellular matrix components. And we can do this sort of in some short-term experiments where we just look at the effect on gene expression, but we'd also like to develop a system where we can load the hydrogels over a period of maybe a few weeks and see if we get a significant accumulation of extracellular matrix components and see if we're really getting a functional production of these components. And if that is the case, we can actually also measure the mechanical properties of this construct, and if there's a significant amount of extracellular matrix produced, it should become more mechanically robust. And then eventually we'd like to identify some downstream mediators, maybe using inhibitors of some putative targets such as these.
I have a question for you. I mean, I don't know how easy it is to molecularly manipulate meniscus cells. I'm expecting it's probably pretty tricky, given all the things you said. Would it be easier to upregulate expression as opposed to knockdown? Because that's the other way of testing. I just wonder, if you had that in your list of things that you might do.
Yeah, I think that would be interesting to do eventually. As far as how difficult it is, for isolated cells, and I'm going to talk about the system that I'm using for the knockdown, but we can transduce them with lentivirus pretty effectively, so that allows us to deliver our CRISPR construct for the knockdown, and we could also use that technology to maybe upregulate, and I definitely think that might be something to try. It doesn't really work in the native tissue is the problem. The virus doesn't really seem to penetrate, or it's not effective to cells and tissue. So that's a limitation if we want to do anything kind of in situ, but that's what we use currently. So you might ask, is TRPV4 expressed in meniscus cells? And yes, it is. Not as highly as in cartilage, but interestingly, the inner zone cells seem to express more TRPV4 than the outer zone cells, which kind of makes sense. The inner zone is really where you have the compressive type forces, the outer zone. It may be a different mechanism because there's more of this radial stretch component to maybe the forces that those cells experience. So the system that I've been using to try and knock down TRPV4 is a CRISPR dCas9 CRAB, in which we use a deactivated Cas9, so there's no nuclease activity, and it's fused to a Kruppel-associated box, which is a transcriptional repressor. And then we target our guide RNAs to the promoter region of our gene of interest. And it just essentially just sits down there and blocks transcription of that gene. And we deliver this, like I said, with lentivirus, so the construct does integrate into the host genome and is stably expressed.
Question.
Yeah.
Is there a reason you decided to go with CRISPR interference instead of knockout?
Yeah, so, I mean, the simple answer is because it was recommended by our collaborators who are experts in this type of thing, but I think the advantage, for one thing, these primary cells, it's not something where we're going to get a single clone that's totally knocked out and then multiply that clone. We need something that works pretty well for a lot of cells at once rather than finding one, because we can't, these are primary cells and they don't replicate indefinitely. This system could also potentially be, we could easily make it inducible, so we can kind of turn it on or off, and the same approach is a little bit more tricky, but it actually can be used to upregulate expression too, so that might be something we could potentially use in the future that way as well. Yeah.
Have you tried any of the other compressible CRISPR systems with multiple copies?
I have not actually. Would that be some advantage to that, or?
I'm just not sure. It depends on how hard you're on.
Okay. All right. Well, I'll show you where I'm at with it currently. So I cloned seven test guide RNAs that were all somewhere in the promoter region of TRPV4. And actually all the guide RNAs showed some level of repression of TRPV4 expression. And this was using 100, this is basically the highest dose I could use. We were just hoping to see something. And I think maybe this amount of virus might actually be having some sort of just viral effect on the cell, because even our control shows some. suppression, but actually at a lower dose, we get a really effective suppression of TRPV4 expression, and the control appears to be pretty much unchanged. And additionally, when I test an unrelated gene, we don't really see much of an effect, or certainly not any suppression of expression of KSO1, which is another ion channel, but it's not genetically related. So based on those promising results, I took 3 of the most effective guide RNAs and tested them at several doses. And these results are very promising. We were getting really robust suppression of expression. And so I've yet to maybe kind of find the ideal dose, and it's a little bit complicated since we're delivering it by virus, rather than, it's not as simple as maybe titrating a concentration of a small molecule inhibitor. There's some batch-to-batch variation in the virus production. There's the ratio of viral particles to cells, as well as the volume. So there's kind of a lot of factors, but it looks like it works pretty well, so I'm excited to start using this. Hopefully soon, to, as I said, measure the effect of this knockdown on intracellular calcium, as measured by fluorescent calcium dye, as well as embedding these cells in some sort of a hydrogel system, applying some dynamic load, and see how that affects gene expression. And hopefully we'll see some differences in the accumulation of extracellular matrix components and mechanical properties. So to that end, I've been working on testing a variety of hydrogels for this purpose. And for similar experiments in chondrocytes, 2% agarose has been used, and it seems to work pretty well for chondrocytes, but in our experience, not so well for meniscus cells. And one of the reasons for this is that it breaks down over time in culture. And chondrocytes, or at least when you use chondrocytes from young pigs, at least, they produce a large amount of extracellular matrix. It essentially replaces the agarose, so they hold up over time. But the meniscus cells just aren't as active. So we're looking for a material that we might be able to use for some more long-term culture experiments. So the ideal material would be mechanically robust, meaning both that it will hold up over time in culture, but also that it has to be stiff enough to really transmit these loads to the cells. So the chondrocytes and meniscus cells, they form sort of a pericellular matrix, which is a little zone of collagen around the cell itself, and it's quite a bit more stiff than the material. So if you imagine a kind of like a stiff ball in a soft material, when you compress the material, only a percentage of the force is going to really be transmitted through the pericellular matrix and actually have an effect on the cell. So it needs to be stiff enough to really effectively transmit these forces to the cell. And it's hard to say exactly how stiff is stiff enough, except that we know that 2% agarose works for chondrocytes. So for my purposes, I've been defining that as at least as stiff as the 2% agarose. Obviously, we need our cells to be viable within this hydrogel construct, and we want them to maintain a differentiated phenotype. So one of the issues with working with primary chondrocytes and meniscus cells is that they pretty rapidly de-differentiate in